Okay, it's 12 o'clock, right at the hour. So let me begin by welcoming everyone who has just joined us for this webinar. My name is Paul Ndebele. I'm part of the Bioethics Interest Group at the George Washington University. I'm also part of the Office of Research Excellence. So on behalf of the Bioethics Interest Group and the Office of Research Excellence, I would like to welcome you all to this webinar that is focusing on ethics and statistics education. So this basically is a forum for exchanging information and ideas about public health ethics. And today, particularly, we're focusing on the area of statistics. Today, we are privileged to have two great panelists in the form of Dr. Ramila Raman, who is an associate professor of biostatistics, health policy, psychiatry, and behavioral sciences and nursing in the Department of Biostatistics at the School of Medicine at the Vanderbilt University. Thank you very much for accepting our invitation. And our second panelist is our own Dr. Kate Crandall, who is the director of the Computational Biology Institute. He's also director of the Genomics Core, which is under our School of Public Health. So thank you so much, Kate, for accepting our invitation. In terms of how we are going to proceed with this webinar, we are going to move to the panelist presentations as the next item. We're going to have Dr. Ramila Raman presenting first, followed by Dr. Crandall. And then after that, we're going to go through a moderated question and answer session. And then again, invite our two panelists for some closing remarks right at the end. So in terms of our questions and answer session, we can actually use the Q&A function at the bottom of our screens. So as our two panelists give their presentations, let's feel free to send in questions and also comments. We're going to go through some of those. Just as an update, the next meeting, the next webinar is going to be on the 15th of November, and it's focusing on diversity in research training. This again is a very, very important topic that warrants our attention as individuals who are interested in ensuring diversity and inclusivity in research. So these are the panelists who have agreed to be part of that webinar. We're going to send information by email shortly during the coming days. So on behalf of the Office of Research Excellence, once again, I would like to thank you all for participating in this webinar. And I just want to emphasize that for our students in the School of Public Health, they can actually submit their professional enhancement activity forms to our big email address so that they can receive the required signature. So be assured that this webinar counts in terms of your PE requirements. So now, without wasting time, I am going to invite Dr. Raman to share views with us on this interesting topic of today. Thank you so much.
Thank you so much, Dr. Ndebele. Let me try sharing my screen now. Just a moment. Can everyone see my screen properly?
Yes.
Oh, awesome. Thank you. It's a great pleasure to be here today. And as mentioned, my name is Ramila Raman. I'm an associate professor of biostatistics at Vanderbilt University School of Medicine. I'm a collaborative statistician, so I work with various groups across campus to answer interesting questions. I work with the Department of Children's Services to evaluate quality control for children in state custody and so on. So I've certainly come across a lot of situations where I wondered, where should I stand on this ethical predicament? Like, do I say yes to this? Do I say no to this? Do I take so-and-so decision? So this is a very broad field for me. I wondered where I should start first, and I figured that I should probably focus on a recent paper of ours that we published early this year. And it brought me back to think how I started getting interested in statistics and ethics. And I remember the years when I was a graduate student, my advisor once came to me and asked me if I'd like to go along with him to an IRB meeting. IRB basically meaning Institutional Review Board meeting, where there are committee members who decide on whether or not to approve a research study based on the ethics. And I attended that meeting, and it was fascinating to me. I enjoyed being there for the 3 hours that they discussed this one particular study. It was just a common question that they all had in mind, but they went back and forth till they decided or came to a common decision on what to do. And oftentimes with ethics, there is no black or white answer, a yes or no answer. There's no perfect answer, but there's always a better option. And we always strive to make that better option. So after graduate school, I came to Vanderbilt, and I also had the opportunity to serve on our IRB board over here. And I happened to meet the last author of this paper, Dr. Hyatt, who is a statistician who also serves on their IRB board at Georgia State University. So we both started to talk about the different aspects of ethics and statistics. And we both mentioned that there are lots of resources and books out there that talk about how ethics applies to statistical practice. So when you're a practicing statistician or a biostatistician or a quantitative practitioner, there are lots of discussions on how do you talk about p-values, how do you conduct your analysis, and all those decisions. But there aren't many resources on how instructors can teach statistics specifically in an undergraduate setting. And I'll talk about why we decided to focus on undergraduate coursework on introductory statistics. We looked at the American Statistical Association guidelines, the International Statistical Institution guidelines, and these are ethics guidelines for practicing statisticians. They both echo a common set of principles and shared professional values for the practicing statisticians. So there are lots of resources there on what to do, what not to do. But once again, they graze along the topic of what an instructor should teach their students about ethics, specifically introductory statistics. So why introductory statistics? So the literature has shown that about 35% of the universities that offer some sort of quantitative program, graduate or biostatistics programs, require an ethics course for at least some students to obtain the degree, which means that graduate students are often going through their graduate coursework with some discussion about ethics and statistics. Additionally, these are graduate students that we're talking about. They work with real data all the time. So when you start working with real data that you have to consent participants for and so on, you have to go through this coursework called responsible conduct of research. So they have this discussion about ethical principles and training materials. So they're exposed in different ways to ethics and statistics. But there are many statisticians in the field who feel like you're restricting ethical responsibility and you start teaching these students only during graduate coursework, and you miss those 4 years of undergraduate coursework. Those are opportunities that you could be having these discussions with these students. And ethics is sort of considered to be like a muscle that you keep exercising and you make it better rather than something that you instantly get or not get. So a lot of this has to do with how statistics education has evolved over time. So when we think about introductory biostatistics or statistics courses for non-quantitative majors, during the undergraduate years, 4 years, they might take one single statistics course, right? That's it. They're scared to come to that class. It is mandatory. They take it, they graduate, and they're excited about it. I'm a very small person. I'm not very intimidating, but when I walk into a class of 200 students, they're not happy with me, right? They're fearful. They're thinking about formulas. And most of this is because in the early 1990s, the focus used to be purely on formulas. I would give them a cheat sheet. They would look at that. They would plug in the numbers and they would get the answers. And that's how they used to get graded. But in the early 2000s, there was a major shift in the way statistics was being taught. You talked about concepts, you talked about reasoning, critical thinking, and more importantly, you emphasized real data. And it is important for me to note that in introductory undergraduate classrooms, there are different types of students there. There are students who may end up being producers of data, that is, students who may end up actually generating reports, analyzing data. And they may also be students who are consumers of data. So they might be physiotherapists, nurses, and they might not analyze data anytime in the future, but they're consumers of data. They read news reports, they read clinical trial reports or journal articles. So it is important for them also to understand the ethical implications of the statistics that have been generated. So talking about the evolution of how statistics instruction has changed, the emphasis on real data ended up being both a blessing and a curse. A blessing because you were able to engage students more in statistics. Students were actually interested in statistics, and you can see that by this big rise in statistics professions over the last 10 years. And it also ended up being a curse because students started using real data, but instructors did not ever emphasize where the data came from. Did the participants consent to the use of data and all the ethical implications of the use of that data? Undergraduate data science programs are particularly good at this. They've recognized this and have included ethics as one of their core topics in their curricular guidelines. And it might be because of the nature of how data science works and how statistics works, right? Data science, they mainly mine the data from the internet. And so there are many ethical implications to that. So they've made it a point to include it as one of their core topics. So when we think about ethics and statistics, many papers can be written about it and have been written about it. Many books can be written about it. And they mainly focus on the statistical problem-solving process, specifically for producers. Once again, like I mentioned earlier, how do you define a p-value? How do you think something statistically significant, and so on? And we looked at a couple of guidelines that are provided to practitioners. One is the ASA's ethical guidelines for professional practice. The other is a guideline called GAZE. It stands for Guidelines for Assessment and Instruction in Statistics Education. Now, this is a guideline that is widely used by instructors of introductory statistics courses. And they talk about how you need to use real data. But once again, both these guidelines do not explicitly mention how you can incorporate ethics into statistics education. And we do feel like it is very important to weave ethical themes into introductory courses, particularly regarding the data collection pipeline, given the use of secondary data sets. So I'll provide a few examples. These are a few toy examples that are commonly used in many, many books and statistical courses. One is a Titanic data set, and the other one is a credit card approval data set. And these are secondary data sets, and ethical concerns appear even if we are using secondary data sets for our analysis. When we provide these to students, it is very natural for students to be eager and jump right into the analytics and not think about ethical issues on data provenance or where the data came from. And I think it's important as instructors that we lead the discussion on the ethical implications of using these secondary data sets. So here are a couple of prompts that I commonly use in class. One is, are the data sufficiently anonymized or old to be free of ethical concerns? Do we have permission to use these data sets? And when you think of the Titanic data set, we all use it, right? I haven't come across a single undergraduate course that hasn't referred to it, or many books have the Titanic data set. It's commonly used to fit linear regression models. But one thing nobody thinks about is, did the participants consent to the use of this data? Many of the participants were deceased in the Titanic, so there is no way they would have consented to the use of these data. But of course, these are older times when consent wasn't exactly defined. So that is something to think about. The other thing to think about is, did the participants consent to the data being used specifically for that research question? And I commonly provide this example of the Havasupai lawsuit. The Havasupai were tribe members who provided DNA samples to researchers specifically to study diabetes. And they realized later on that their DNA samples were used to study different health conditions, not just diabetes. And a lawsuit was brought about by them, and it was finally settled. So this issue of whether or not participants consent to the data for a specific hypothesis or a broad nature of research is still a question that is being considered. The other question that I consider is, do the data have identifiable information? We all have a right to privacy, right? And a common example that I use is the Sweeney-William Weld example. William Weld used to be the governor of Massachusetts, and he made public a lot of information for researchers, and he assured the public that it was de-identifiable information. But Sweeney was a researcher who went in, got that data set, combined it with several other publicly available data sets like census data. And she was able to identify exactly a couple of people. And that opened the eyes to what identifiable and de-identifiable mean, right? A data set may be de-identifiable when it stands alone, but when you combine it with different resources, it could be identifiable. This was one of the examples and the reasons why the HIPAA started coming about. And it shows me that we all make mistakes, but it's important to learn from our mistakes and make things better. Other few prompts that I ask are, were the variables collected in an accurate manner? Were they rigorous? Should we analyze data if we do not know how the data were collected? Oftentimes, we have publicly available data like the census data, enhanced data, and so on. It is very easy for us as researchers to say, hey, there is a data set available there. Let's tackle this question. But it's also important to understand how they were collected. Were they collected through a survey means? Was it a randomized clinical trial? What was the design of the study? And so on. So in order to conclude, I would like to say that there have been so much societal changes over the past 20 years with respect to technology that have provided us the ability to obtain lots of data. And it can be both a curse and a blessing, but it's important to bring about a conversation about ethical aspects of using such data. And I'd like to end my conclusion with this quote that I think is really powerful by Gardner from 2011. And I'm going to read it verbatim. An issue that may be overlooked is, how do we know what the data are? They do not simply appear out of nowhere, neatly organized and arrayed for analysis. I read that and I remember the countless times where I've just given my students the Titanic data set, right? As though it just appeared from nowhere without providing much context to them. So once again, as instructors, I feel like it is our responsibility to bring about these conversations so that students who may not end up being practitioners, but even just consumers, can be educated about ethics in statistics. That's all I had for today. Thank you. My contact information is provided here. So if you guys have any other questions after today's panel, I'm happy to answer them as well.
Thank you very much, Dr. Raman, for this brief presentation. By telling us how you got involved in statistics, you also even showed us the close linkage between ethics and statistics. You also described the evolution of statistics education over the past decades. And more importantly, you raised so many questions that relate to the ethical implications of using various, the data sets obtained from various sources. And of course, you emphasized on the importance of weaving ethical themes into introductory statistics courses. So thank you very much for sharing your views on this topic. So now we're going to move over to our second panelist, Dr. Crandall. Let me also encourage our participants to please submit your comments and also questions through the Q&A function at the bottom of your screens. Thank you.
All right, are you seeing the full screen or the presenter view screen? The full screen?
Yes.
Perfect. All right. So good afternoon, everybody. Thanks for that great start, Romila. That was really right on and totally different from anything I'm thinking about, because I actually am more of a computational biologist, bioinformatician than a true statistician. Although I do have a master's degree in statistics, I do more statistics applied to omics kinds of data, which leads to all kinds of interesting and different kinds of questions. Although to a certain extent, they're fairly similar. And you can see from these multiple hats that I wear as director of the Computational Biology Institute at GW, the Genomics Core, the Clinical and Translational Science Institute, Informatics co-director, in our health data science, our new health data science PhD program co-director. We've got this intersection of omics and statistics and health going on all the time. And there are lots of, for me, I think it's really a ripe area for investigating questions of ethics, statistics, omics, and then we do all the educational pieces to this as well. So I really appreciate, especially the last presentation on how to integrate some of these things and think about getting these into the classroom. Because frankly, I don't do much of that at the moment because I'm really not sure where I can even start. So you'll see at the end, I'm going to make some proposals to the entire group here and hope that we can work together on trying to figure out some of this stuff. So we can think about statistical significance as the sort of quintessential ethics and statistics problem, right? And Romela kind of talked about that as well, right? That we talk about p-values and what p-value cutoff should we use and why should we use that? We tend to use 0.05, right? Because it's a certain standard deviation away from a normal distribution. But other than that, there's not a real good justification for it. I have seen in human genetics, especially, people shift that to a p of 0.1 because, especially in the age of genome-wide association studies where you're trying to identify variants, genomic variants associated with disease, you don't want to miss things. So you're willing to err on the side of false positives. So you change that p-value. And I've seen it go the other way, right? Where if you're making, health decisions, treatment decisions on statistical cutoffs, then you're maybe not as willing to err on the side of false positives. So you really want to slide that p-value over. So while on average we think about 0.05 as the be-all and end-all, I think it's important to recognize that is relatively arbitrary, and we can slide that scale depending on in a lot of cases, what are our downstream actions? Are we gonna do more computation downstream? In which case, let's have a bunch of false positives and not worry about it, and we can sift through those out the end. Or are we going to do a bunch of molecular biology work to validate things? In which case, there aren't patient issues going on immediately, but there's cost going on. And how many false positives do you want to sift through at some finite cost to your research program to move forward versus the extreme end of having actual treatment decisions based on this impacting human health directly, and how many false positives do you want to deal with there, right? So I think thinking about p-values, about statistical significance, and the biological significance relative to the statistical significance is really an important issue. One of the things that we always need to keep in mind as researchers is the problem of multiple comparisons and how to correct for that. And that gets even more accentuated in the age of omics. So omics is a beautiful age with tons of data and really, really minuscule p-values because you have millions and millions of observations. So you can do a lot of comparisons and still get statistical significance, but you have to start wondering about, where cutoffs. 0.05 is out the window, right? I mean, you don't even start there. And you have to kind of think a lot more seriously about whether p-values are even what you want to look at, or false detection rates, and where reasonable cutoffs are, but using that same concept of what are the downstream activities and how do those interact with the decisions you're making on the proximate data set and make adjustments accordingly. There are, in my view, in my world of omics, there are some really interesting questions out there that were, because this is relatively new areas for lots of us, there aren't established protocols. There aren't, the American Statistical Society has not come out with guidelines. Nobody's frankly come out with guidelines on how to deal with omics data. We do metagenomics data, which is basically taking a typical study in our genomics core is doing, for example, a gut microbiome analysis, where we take a stool sample, we sequence the stool sample from an individual, we try and collect, 6 million 150 base pairs snippets of DNA from that stool sample. 3 million of those are from the host, and 3 million of those are from the microbes. And we have algorithms that partition those. And then we have options, right? We can investigate what's going on with the host and the host genetics. We can investigate what's going on in the microbes, which is, in microbiome studies is what you're particularly interested in. But sometimes there are variants on especially if it's, instead of metagenomics, it's metatranscriptomics, it's gene expression data. So you can get the expression profile of the microbes, but the gene expression profile of the host as well. So you can look at that interaction and ask questions about how the host immune system is interacting with the expressed microbes, or in the genomics case, how the host genetic background is impacting or interacting with the microbial population in the microbiome. So, and of course, we do all these comparisons using statistics, using typically machine learning approaches now, but it raises some really interesting questions, because as you survey the host genetic variation, you could, and you often do, find genetic variants of known health significance, right? So the one of the many ethical questions here is, what do I do with that information? One of our microbiome research studies is with a collaborator in the School of Public Health, who is, testing artificial sweeteners on the impact of the gut microbiome, and had stool samples from students at GW, bring those stool samples in, and we do this metagenomics. If we detect genetic variants that are known associates with disease in those students, in those healthy individuals who are contributing to our research project, is there some obligation or not to report those back to those individuals? We are not physicians. We're not in a good place to do that sort of thing. When we publish our data, in the age of open access and open data and NIH and NSF initiatives to do open science and make data available, do we submit all the metagenomic data, including the host component? Because I'm here to tell you, Romela had this great discussion about, how de-identified data isn't as de-identified as sometimes we think it is, right? Especially when you start combining it with other data sets. Well, guess what's the ultimate and non-de-identified data is genomic data, right? We can de-identify it. There's no name on it. We can disassociate age and whatever other demographic variables we're collecting from the sequence data. But it's genomic sequence data in the end of the day, right? So it's not, and we've all seen these kinds of reports. It's not too difficult for a reasonable bioinformatician to capitalize on variants in that random or that anonymized host DNA and combine it with data from, for example, ancestry.com, and start really getting down and dirty in terms of who these individuals might be, right? So there's this conflict of interest between being able to have reproducible science, and being able to combine studies across science, because meta-analysis is an exceptionally powerful way to do science. If we're doing this study at GW and somebody else is doing a similar study at Vanderbilt and somebody else is doing a similar study at the University of Michigan, and you want to put all three of these together, you want all the data to investigate. But how do we do that in a reasonable way? Do we submit all these data to GenBank in the sequence read archive so they're open access to everybody, or do we do some filtering of the human reads and then just present the microbial reads, which makes the combining of data sets more difficult and makes some of our results, especially with respect to the host, irreproducible in terms of the science, right? So there are some real significant questions, I think, around data availability, meta and subsequent meta-analyses, with especially the omics data in how we're analyzing these kinds of data. And of course, here at GW, we teach a genomics course, we teach a bioinformatics course, and there's ample opportunity to integrate some of these ethical questions into these kinds of courses, which I frankly don't do and don't know how to do and would love to do. So I'm looking for groupthink on this from y'all out there, and hope you can help me figure out how to do this. There have been some recent papers, though, talking about exactly these kinds of issues, setting up the difficulties. And here's one that just came out. You can see it's came out October 2. So just this month, Curtis Huttenauer and his group. Curtis is a faculty at MIT, does a lot of the same kinds of things we do, software development for microbiome analyses, and lots of applications of that software in a wide variety of microbiome studies in humans and other organisms. And the paper details, and I'll pop, once I'm done with this, which will be soon, I'll pop these references into the chat so you can all link directly to them. But challenges and opportunities in sharing microbiome data and analyses, right? So part of it is not only being upfront and obvious about the data and whether you can do that effectively or not, especially when the data are coming from humans, but also about your workflows and what you're actually up to in terms of the analyses. In the day of electronic publishing, where page numbers seem to not impact the cost of publishing papers at all, somehow our journals are still getting more and more streamlined with respect to methods, to the point that you can't actually replicate almost any science. Take any paper in Science and Nature and try and replicate it from the science. Good luck with that, because they're just not detailed enough. And especially in microbiome analyses and any kind of computational analysis, having, the actual scripts that people are using is really the ultimate in reproducibility of science, and we just aren't there yet. And if that is our ethical standard for analysis, then we need to develop the capacity to abide by that ethical standard. And I think we're just not there yet. And then share that expectation as reviewers, as journal editors, and as authors of these papers, as researchers on all sides of this, and make sure that we are expecting that of ourselves and of our colleagues. And if it's not there, we need to call them on it and make sure that those things are there. The other example, microbiome analysis is part of our work. The other work is a lot of HIV sequence analysis. And this is one that drove me crazy for a long time, because we especially want to do a lot of combined analyses of HIV. We do HIV analysis in Washington, D.C., and I'm sure Vanderbilt does it in Nashville, Tennessee, and all around Tennessee, and Harvard does it in Boston, and UCSF does it in San Francisco, and Emory does it in Atlanta, Georgia. And at some point, you really want to combine these data, right? And you want to make inferences from across these data and see what's going on in the US. And then this is replicated across the world, and you want to be able to combine those data. And yet if people aren't making their data available, is problematic, especially around, this paper is really focused on drug resistance, right? One of the big issues with HIV is the evolution of drug resistance of the virus. And we have a lot of data. In fact, there's a database at Stanford that accumulates all the data on drug-resistant variants. But the database is only as good as the data that you put into it. And we've developed, and other folks have developed, machine learning approaches to predict drug resistance from a set of sequences. But those predictions, any machine learning approach, is only as good as the data that you train it on. And if the data you train it on are limited because people are not sharing the data, that's problematic. But of course, there are good reasons that people are not sharing their data. And even though it's viral data that you'd think would be pretty benign in terms of sharing, because unlike the microbiome data, there isn't a host component to it, there are you can establish transmission linkages, which is why we do some of the sequencing in the first place. You know, we try and identify transmission linkages so we can better direct public health interventions to specific geographic areas with specific demographic information, right? I mean, in public health, we have needles and we have condoms, and you need to know who to give needles to and who to give condoms to, and where those groups are where transmissions are occurring, or both, right? So surveying the virus for its sequence data and making those inferences on the virus is infinitely more reliable than asking individuals, who have you had sex with lately and where do they live, right? You can imagine the information you get from that. So we can do molecular epidemiology on the viral sequences and establish transmission patterns and linkages, and then target interventions accordingly. But those same data people can use to, and have used to disparage individuals that are in transmission networks, to present legal action against individuals in transmission networks, right? So there are all kinds of legal and ethical issues associated with the data, even when it's just from the virus, because of the ability to establish transmission networks. So this paper kind of is talking about that kind of trade-off and how we might move forward with getting that important drug resistance data that we need to combat HIV, and yet still hopefully keeping some reasonable privacy with respect to transmission patterns, et cetera. So lots of great questions here, and importantly, lots of great opportunities and research opportunities to do this research, right? So this is from yesterday's email, right? This just came out yesterday, 16th of October. The Ethical and Responsible Research Program is out there to foster ethical research practices. NSF is giving grants in this area. So if there's a group of faculty and students in this partnership here that are interested in applying for grants to do this research around the ethical practices, I think this is a great opportunity, right? There's funding in this because it is important. The National Science Foundation understands it's important. NIH understands it's important. We all think it's important, which is why we're here. And we can, I think, make some headway, both in proposing some concepts and then doing the research as well. So that's what I have, and look forward to opening this up to a broader discussion here.
Thank you very much, Kate, for raising lots of difficult questions, particularly around metagenomics studies. You raised the issues around the feedback of findings to the individuals who have provided the specimens, particularly those findings that have health implications, the individuals. You also raised further the issues around identifiability of individuals, when we think that we are actually using anonymized samples. You also raised questions regarding data availability, issues around the sharing of data, and of course, lots of issues around privacy. So thank you very much for giving us some stuff to think about. So now we're going to proceed into the question and answer segment. And I want to encourage our participants to send in their questions and comments through the Q&A function at the bottom of your screen. But right away, there is one colleague who has presented a question which reads, what would be the best option if I discover at a later stage that the statistical data I have been analyzing have been collected in a manner that violates the participants' rights? More so, that the analysis I could have done at that point would be quite useful to the conclusion of my study. So this is kind of a tricky question. What would be the best way to proceed? We can start off with Ramila.
That is a very good and a very hard question. You know, you have to try to figure out the greater good of mankind or an individual's rights, right? But we have never been in a situation like this before, but typically the institutional IRB will provide you some kind of guidance. There have been situations where I work a lot in clinical trials, and there have been situations where participants have withdrawn from the study, but we still have had that data. And oftentimes when we have found that, we have gone ahead and deleted it, documented it. So for the most part, I do feel like we have to be honest about it. It is also not an individual's decision to take, right? Oftentimes we work with collaborators, and I think it's very important to have this discussion with a lot of people to figure out where we should stand regarding this.
Thank you. Yes, Keith.
And I just say that there's, you know, there's institutional help with this sort of thing, right?
Yeah.
So take advantage of your institution, your compliance officer, your ethics review board, and report such findings first and foremost, and then seek their advice and guidance on next steps. Would be my I try real hard not to get in trouble. So
yeah.
Talking to people whose livelihood it is to worry about these things is always the best first approximation for me.
So this is basically a difficult position that one would find themselves in, where you now realize that you're actually working with this data set, and yet at the same time you realize that the findings are important. This reminds me of some of the horrible studies that were conducted, for example, during the Second World War by the German scientists, right? And yet, still up to now, we refer to some of those findings, because the question is, are you then going to dump all that data? If you dump the data, it actually means that now another study has to be conducted, which means some more resources have to be pumped into that same study. So this is a very, very difficult question, and at times some might have to proceed and publish after acknowledging that there are some issues related to the way the data were collected. But I appreciate the fact that you're actually trying to emphasize here that you are better off not putting this burden on yourself as an individual, but working with others, with your IRB, to make sure that they can assist you as an investigator in getting to a decision that you might feel good about at the end of the day. Thank you so much. I want to move on to the second question. This again relates to the issue of the origins of the data sets that we use at times. So this question is about someone who has no idea about how data were originally obtained. So how they have no idea about how the data set was originally obtained. So they're asking the questions, is it possible that there could be some ethical questions about the accuracy of the accuracy or the inaccuracy of the conclusions that are going to be reached based on that sort of data set? Let's hear your views.
Certainly any statistician or bioinformatician will tell you that the first thing we worry about are the data origins and the data quality. And if you have no idea of the origins, then you have to think really seriously about how useful any analysis of those data are going to be anyway, right? There are some traditional data sets that are out there and available, but you should be able to find origins of those and be able to document where those come from. If you can't, then I would say move on to a different data set, because that's really problematic. Secondly is never trust data that you're given no matter what, right? And start by doing QA, QC on the data to begin with, because researchers do strange things, and you never quite know what kind of data you're going to get. So it's really important to worry about contamination, worry about mislabeling, worry about anything and everything associated with the data, as if you collected it yourself and this is your first look at the data, and you want to make sure, because everything you do downstream is dependent on the quality of those data. So it's, the garbage in, garbage out adage is right there, and it's not, frankly, worth your time and effort to work on a data set that has no known attribution and or has problems with what's with quality and whether you can trust the data or not.
Yeah, I would divide that question into two parts. One is, for teaching, I think it's fine to use story data sets as long as you have a discussion with students on, I have no idea where this came from. Take the results with a grain of salt. But when it comes to publishing, I feel like researchers have some sort of moral obligation, right, to make sure that the data is accurate, the variables are rigorous, because the conclusions depend on that, like Keith just mentioned. So I treat it with great seriousness. And even when we share data, I think the documentation is often longer than the data itself. We document every single decision, how a specific variable was coded, how it was collected, what has been changed after the primary manuscript was submitted, and so on. So I think documentation is key in those situations, and it is very important to have quality data.
Thank you. I want to emphasize on the point that you both raised about documenting the sources of data, so that at the end of the day, you are able to respond to those questions about the inaccuracies of your conclusions or findings, right? So thank you so much. Kate, you mentioned issues around privacy when you get to the issue of sharing the omics data sets that you work with. So what are some of the steps that you take as an individual scientist to ensure that you continue to protect the individuals who are part of those huge data sets?
Well, the first thing we do is, we de-identify the data. So we have metadata associated with the sequence data. So, we know sex and age in almost every study. They usually have some patient ID, P1 through whatever, right? So that's generally, we typically don't know any names by the time we get samples and data. But we do have metadata that's important for the analysis. We've done a lot of SARS analyses where we're getting clinical data. So we're getting, drug treatment information. We're getting BMI information. We're getting, health outcome data. So those are all very sensitive data, and they all have dates associated with them. So when we publish those studies, we do summary statistics on especially the demographic and clinical data. So we're not reporting out full data tables. And then for the sequence end, we partition any host data out and then submit the microbial component to GenBank, so that's available for reanalysis, right? But at the end of the day, it's still this trade-off between keeping the privacy and allowing reproducibility in science, because still, if people don't have that raw metadata matrix, there's no way they can reproduce my analysis. And if they don't have the host component of the microbiome, there's no way they can redo some of the analyses that we're doing. So it's inherently, as soon as I hold back some of that information for privacy concerns, my science becomes non-reproducible, at least off the shelf. And it becomes non-combinable with additional data sets. So if people do reach out and want to combine data sets, then we have to have all kinds of conversations, further IRB, before we can share those kinds of data, right? So it's complicated because there is this inherent trade-off between reproducibility and open science, which is where we're all trying to move to, and yet being reined in by significant and legitimate privacy issues.
Great. Ramila, your thoughts?
Yeah, we share data a lot. I work with a big ICU group here, and our statistics department motto is that we need to be transparent and share data. And there are guidelines out there on how to de-identify data. So for example, if someone is older than 90, you should just censor it at 90. If you have zip code data, you should be more restrictive with the age and so on. We try to do all that, but at the end of the day, it is still a hard decision, right? Because when you send your data to someone else and the age is censored at 90, they cannot analyze the data like how we would be able to do it. So it limits them in a lot of ways. So it's always a hard decision. Once again, we go to our IRB, we talk to them about it, whether we should share a de-identified data set or a limited data set, where the researchers have a few more variables than we do. We always make sure that our data is stored in a secure place. So we do not have paper copies anymore. It's all electronic, but there are multiple steps to go through that. We do not have identifiers in a database that is placed in a separate section and so on. So we have safeguards. The issue occurs when all the safeguards fall at the same time. So we try to make sure that there are a couple of people checking it. And when we send data out, we are very, very careful about it.
Great. And Kate, you mentioned an issue related to questions around the feedback of findings, particularly those ones with health implications to individuals. So what are some of the situations that we have had to deal with related to feeding back of findings to individuals?
Yeah, this is more of a hypothetical because we do a lot of metagenomic data. So we haven't purposefully, we have not done mapping of the human component of the metagenomics reads to variants of known health impact. There are a lot of known cancer biomarkers, for example, that you could test against. I mean, this is what 23andMe does, right, on their health side commercially. It turns out we can do the same on a study side, but we don't because we're not quite sure what we would do if we discovered variants that were of health significance. So, but there's an ethical question there of just should we be doing that? I mean, is that something that would benefit individuals who participate in these studies to have that information? And are we, complicit in not providing that information when we could do? Or is it, if we do it and it creates, anxiety that would otherwise not be there, then is that problematic? I mean, 23andMe kind of wrestles with the same issue, although there people are paying for that information. So presumably they want it, whether that creates anxiety or not. But with us, it's kind of a spin-off of a study. So it's a little more up in the air, I think, from our perspective. So we haven't done anything. We frankly don't want to do anything. And if we did move down that path, then we definitely have to have basically a team of individuals, including some good ethics people, some good medical people, who could better, genetics counselors who could better inform individuals of what the significance of these discovered variants might actually be. And to me, my view is always, it's better to have information than to not. And if I had variants for prostate cancer, I'd want to know about it sooner rather than later, so I can do appropriate testing earlier rather than later, and make sure that I'm keeping tabs on things, right? So, but people don't necessarily share that particular view. That's my view. Some people don't want to know and are happy to not know. So it's, I don't know, it's a tricky thing.
Right.
I'm here to generate questions, not answers.
Yes, Ramila, any thoughts?
I remember a situation like this when I served on the IRB. And one thing that the IRB suggested was we write in the consent document and give the participants a choice on whether they would like to know the results if the data were to be used for a hypothesis that was not mentioned. So the approval was very broad, and I'm not sure if many participants consented to that, because I think it's an individual's preference, but I certainly feel like IRBs are moving towards a direction where they ask the participants, do you want to know? Would you like to be communicated with if we found results that may pertain to your health condition? So,
thank you. That's a great option. There is another question. We'll take this as the last question. Can you please discuss the ethical considerations around data sharing and code sharing associated with publications? How do you balance the need for open data and code with intellectual property?
I think that is hard. Once again, none of this is easy, but there are websites out there. So I work in clinical trials, and it is very important to me personally, as well as a department and a university, that we be open and transparent. And there is a website called Open Science Framework where you can publish code, you can publish metadata and all of those kinds of things. We have tried to do that. The hard part is publishing data and depositing data somewhere. So I do feel like for that, they have to come to us and ask. We need to feel like it's valid. We would not be publishing the participant data just on any public website, right? That is a little dangerous. But we do publish code and all of those kinds of things that might be necessary to reproduce the analysis in some other data set.
Well, there are some good examples of sort of modified open access examples, one of which is the Cancer Genome Atlas, where you have both metadata and gene expression data housed in something called dbGaP, where it's open access in the sense that in theory any researcher can get to it, but you have to go through some hoops to get there. You have to go online and submit a proposal, tell NIH why you're interested in analyzing a particular data set. You have to get sign off by your compliance officer, your research officer at your host institution. You have to verify that you have appropriate facility capacity for downloading the data and storing it locally to do analyses. And the other example is in the COVID pandemic, NIH NCATS established this thing called N3C, the National COVID Cohort Collaborative, where this was sort of the inverse of that, which is instead of having all the data at NIH and then getting permission to go in and access the data, download it, and analyze it in my own backyard, here it's a data enclave. So you still have to go through a proposal, you have to go through credentialing, you have to get approved, but then you still can't download the data. You go into the enclave and do your analyses there, and then you can export your results. So there are different ways to do this to add extra layers of protection that I think are helpful. And then to the last question, you ended this with respect to intellectual property. And that's an even trickier thing, right? Because there's the last thing you want to do is do open access of the concept, your secret sauce that you want to develop a company around, right? I have done that. We developed a company around an algorithm for microbiome characterization, and we successfully developed a company on this, got NSF SBIR money to do this, turned it into a real company that made money and sold it to LabCorp. But and I can tell you that throughout the 4 or 5 years of this company's existence, when I was running it, was the constant question from investors of, we published our algorithm open source, of course, and because we're academics, it's all on GitHub, you can go grab it. And their question was always, well, what prevents anybody else from getting that and doing the same thing? And my answer was, well, nothing. But of course, we took it into the company, and on the company side, we really reinvented the whole thing and made it much better and more user-friendly, and did a lot more machine learning and put in a lot of other things that help physicians think about the impacts of the microbial diversity that we're observing in their patients. But at the start, we couldn't do much. So you have to think carefully about how much you want to protect intellectual property versus get publications and do open science, because they are frankly incompatible. So if you do have desires to do entrepreneurship and develop intellectual property and work through your tech transfer office at the uni, then you need to hold some things closer to the vest to go down that line, would be my advice.
Great. Thank you so much for all those wonderful questions. I see there's another question there related to data security and ethics. Someone is emphasizing on modernizing, but unfortunately, we no longer have the time to discuss this because we are right at the hour. So I'm going to now ask Ramila to give 30 seconds in parting shots. 30 seconds.
I would like to just say something that I tell my students, that ethics is hard, statistics is hard, and when you combine it, things get even worse, right? Especially with this in this era where we have a lot of technology and a lot of data. It is important to make sure that you decide on these things as a group. There are always institutional supports. There are people who support you from the group. There is no right answer to all of these questions, but there is always a better option when you think about it.
Thank you. Okay.
And I'll end mine just by answering Maliha's question, at least to the best I can. And that is, you're right on. I mean, we should be doing, there are automated systems for data management and tracking samples, everything from when you start the study and start taking samples through to analyses. We use some of the software in the genomics core, but institutions on average are loath to put money out for effective software to track things, but they really should. I mean, this is part of what the facilities and administrative overhead should be covering. So I think that's something where Paul, you and our research office can think about, how you can better support researchers by automating some of these and developing software, or not developing, but evaluating software in concert with the researchers and purchasing software to do these sorts of things, so we don't have paper copies of crap hanging around. It'll save you a lot of lawsuit in the end, I think.
Wonderful. So at the end of it all, I'd like to thank our panelists, Ramila and Kate. Thank you so much for taking time to share with us your views on this very, very important topic. I would also like to thank all the participants. I could see, people from various countries. Some joined from Africa, some from Asia. So thank you very much for being part of this wonderful webinar. Let's meet again on the 15th of November, and we are going to be discussing the issue of diversity in terms of research. Thank you so much, and goodbye.
All right. Thanks, everybody.